Okay, good morning, everyone. I also wish to thank you, the organizers, for organizing again this very nice meeting and for inviting me. When I was invited to give a lecture here, I decided to focus on these data, which are not very recent, but they have been recently reevaluated in my lab. And I decided to present this data because there is a story behind that. I think it may be interesting for young researchers who are approaching the topic of ultrasonic vocalizations. So when I was asked to prepare this presentation, I also thought that the speakers before me would nicely introduce the topic of ultrasonic vocalization, so I go straight to the point. And the point is that, as you all know very well, there are several neurotransmitters that regulate the emission of 50 kilohertz ultrasonic vocalizations. Here are some papers from my laboratory and from other laboratories. And among these neurotransmitters, of course, is dopamine. Dopamine, as we all know, is critically involved in the emission of ultrasonic vocalization. These are two very important papers that evaluated the role of dopamine in the nucleus accumbens, actually the role of amphetamine, the effect of amphetamine, that is a dopamine releaser in the nucleus accumbens, and they demonstrated in a very convincing way that dopamine is crucially involved in the emission of 50 kilohertz ultrasonic vocalization. And as you know, dopamine interacts with several neurotransmitters, like, for example, adenosine, glutamate, serotonin, corticosterone, which is not a neurotransmitter actually, with several transmitters that regulate the emission of 50 kilohertz ultrasonic vocalization. So dopamine is a key player in the emission of these calls. And what about the role of dopamine receptors in the emission of ultrasonic vocalization, which is the reason why I am presenting this data today. So here are some previous papers that show that dopamine D1 and D2 receptors may both influence the emission of 50 kilohertz ultrasonic vocalization in response to different types of stimuli, either pharmacological or non-pharmacological. For example, in the very first paper I'm showing here, it was demonstrated that lesions of the VTA or the medial forebrain bundle or the administration of flupentixol, which is a typical antipsychotic drug that blocks D1 and D2 receptors, suppress the emission of 50 kilohertz that are stimulated by a single episode of tickling. In the second paper, there is a study that shows the effect of a D1 receptor antagonist, SCH-23390, and of raclopride, that is an antagonist of D2 and D3 receptors. And they were evaluated against cocaine, and they were able to suppress the emission of calls stimulated by acute cocaine. Here, this is a very complex study that evaluated several D1-like and D2-like antagonists of dopamine receptors, and the effect is always the same, so they can suppress the emission of ultrasonic vocalizations, stimulated in this case by the acute administration of amphetamine. Oops. Here is a study that evaluated, again, tickling. Here is another study that evaluated not the number, but the acoustic features of parameters of ultrasonic vocalization. And here is a more recent study where the title says it all. So that the simultaneous antagonism of dopamine, D1, D2, and D3 receptors in the nucleus accumbens reduces calling behavior that is elicited by rhythmic tactile stroking. So all these studies I have just shown are characterized by the fact that they use the single presentation of a stimulus, either pharmacological or non-pharmacological. So we were interested in the lab to see what happened, what is the role of dopamine in the emission of 50 kilohertz vocalizations that are emitted by rats subjected to the repeated presentation of a stimulus. Since in our laboratory we do studies of pharmacology, we focused on pharmacological stimuli. And we recently published two articles, one about amphetamine and the other about anti-Parkinson drugs. In these studies, we performed a lesion of different dopaminergic systems. In the first one, we performed lesions. I'm sorry, but this is in low definition, I thought, okay, let me explain this to you. So we use in the lab, we use this protocol of repeated intermittent administration of amphetamine or of other drugs. And in this study, we perform discrete lesions of the medial prefrontal cortex or the dorsolateral striatum. The lesions were bilateral. And in this study, we observed that the lesion itself did not affect very much the acute effects of amphetamine, so the effects that were observed after the first administration of amphetamine, but it affected the long-term effect of amphetamine with regard to the emission of ultrasonic vocalizations that were stimulated by re-exposure of animals to the environment where the amphetamine was administered, and also in terms of response to a challenge of amphetamine after treatment discontinuation. In this other study, we performed a unilateral lesion of the medial forebrain bundle. This is a model of Parkinson's disease, the unilateral 6-hydroxydopamine model of Parkinson's disease, which is the most commonly used model for pharmacological studies in Parkinson's disease, and we tested the effect of 3 different anti-Parkinson drugs that are apomorphine, levodopa, and pramipexole. And as you can see, these drugs have different effects on the emission of ultrasonic vocalizations in lesioned animals. It is interesting to notice that these drugs are almost ineffective in eliciting the emission of ultrasonic vocalization in sham-operated animals, so animals that received an injection of saline in the medial forebrain bundle, whereas they are very effective, at least apomorphine and levodopa, are very effective in stimulating the emission of 50 kilohertz vocalizations in lesioned animals. So these studies tell us that dopamine is indeed involved in the emission of ultrasonic vocalizations that is observed in rats repeatedly treated with certain dopaminomimetics, so with either a psychostimulant like amphetamine or with dopaminomimetic drugs that are used in Parkinson's disease. Of course, these studies have a limitation because these are studies that are based in models of dopaminergic denervation. So dopamine is completely removed, at least almost 98% of dopamine is removed in these lesion models. So there is a lack of selectivity for different dopamine receptors. So in these studies, we could not determine if the effects were due to dopamine D1 or to dopamine D2 receptors or to both families of subtypes of receptors. So we questioned what is the role of dopamine D1-like and D2-like receptors in the emission of 50 kilohertz ultrasonic vocalizations in rats subjected to repeated pharmacological treatment. When questioning, it came to my mind this lady that you all should know who she is, don't you? Yes, okay. And I remember listening to an interview in the Italian television when this lady said this sentence is not what exactly she said because I couldn't find the exact quote, but the meaning is that if you don't understand what your data means, or you think your data is useless, put it aside and reevaluate it later. And this is the story that, in my opinion, is interesting for young folks attending the meeting. Look at this picture. This picture is something that I think everybody in this room is familiar with. So all people working with ultrasonic vocalization have a lot of data. This is only part of the data I got in these years. And so thinking about the role of dopamine D1 and D2 receptors, I said, okay, I did some experiments in the past. When I was beginning my research on ultrasonic vocalization, of course, I evaluated the role of dopaminergic antagonists and dopaminergic receptors. But since a lot of paper came out, I said, okay, let's switch to non-dopaminergic transmitters and see what happens. But now, after having done those studies, those lesion studies, I said, okay, let's revisit the data we obtained. And I realized that those data were interesting and they were worth being re-evaluated and they were worth writing a manuscript about. So what we did is we used the very same protocol of drug administration. So we did an intermittent administration of amphetamine, and amphetamine was preceded by the administration of dopaminergic antagonists. We used the SCH-23390, that is a D1 receptor antagonist, or raclopride, which is a selective but not specific antagonist of dopamine receptors, of dopamine D2 receptors, because it has also some affinity for D3 receptors. And the administration of these drugs was followed by the administration of Vicol or amphetamine. We used the dose of 1 milligram per kilogram IP because in our previous studies we have shown and we have understood that if we want to observe long-term modifications in the emission of 50 kilohertz vocalization, I mean sensitization in particular with amphetamine, we have to administer a moderate dose, because if the dose is too high, the animal will not sensitize to amphetamine. So these drugs were administered either in individually or in combination. We gave 3 doses of each antagonist. Both were given subcutaneously, and also we gave them in combination with the lowest doses tested that were not effective on their own. So we wanted to study if there was a synergistic interaction between these two antagonists. So here H means the habituation, treatment means TR means pharmacological treatment. Then we performed a withdrawal in the home cage, a re-exposure to the test cage, followed by a challenge, as usual, we do in our lab. And then we tested at the end of the experiments locomotor activity. We didn't test locomotor activity during the experiments because here we used a setup that did not allow to use locomotor activity counters because there was refraction of the infrared beam. We have changed this setup in some studies, but we are now reusing this kind of setup because the conditioning to the environment is better if we use this setup we use here. So a cylinder that is half black and half striped in black and transparent stripes. So we notice that the conditioning properties of the drug are magnified if we use this kind of setup. So dopaminergic antagonists were always administered in the home cage 15 minutes before amphetamine or Vicol, and USVs were scored. They were recorded on each day of treatment, but since, as you know, scoring is very laborious, we limited scoring to the day 1, 5, 9, and 16 of the experimental protocol. So with no surprise, we found that the administration of a dopamine D1 antagonist suppressed ultrasonic vocalization throughout the pharmacological treatment. So each time the drug was administered, as you can see here, there is a very low number of vocalizations emitted, whereas animals that received only amphetamine emitted a lot of vocalization, and they showed also sensitization to this behavioral response. The same is for raclopride, as you can see here, even if raclopride has I would not say milder because the vocalization rate is very low, but a less pronounced effect compared to SCH, and the combination of drugs leads to a complete suppression of calling behavior. But these data are not surprising because they just replicate what was observed after the acute administration, and they extend previous findings by showing that the role of dopamine D1 and D2 receptors is important even when the drug is administered repeatedly, even when amphetamine is administered repeatedly. Here is the number of vocalizations that rats emitted after re-exposure to the environmental cues, so to the environment where amphetamine and the antagonists were administered. You can see that amphetamine administered alone, rats that received only amphetamine emitted high numbers of ultrasonic vocalization. This is a replication of our previous data. Animals that received SCH at all doses tested emitted some of vocalization, but the increase was not significant compared to amphetamine alone. The same was for raclopride, as you can see here. And what is interesting is that the combination of SCH and raclopride lead to a complete suppression of calling behavior that was significantly lower compared to amphetamine. So this is the re-exposure to the environment in drug-free conditions. So the animals were re-exposed before being challenged with amphetamine. And so the interesting part comes with the challenge. because you can see here is this one is the effect of SCH. You can see that a dose of SCH that was effective during the repeated treatment, so a dose that suppressed the emission, of course, during the repeated treatment, is no longer effective after challenge. So this suppression, of course, is lost after treatment discontinuation, at least for the lower dose of SCH tested, whereas with the higher doses, there is a reduction in the emission of ultrasonic vocalization compared to amphetamine. So the emission is no longer significant as it is after the as it is in rats that received the lowest dose tested of SCH. With raclopride, there is a complete loss of suppressant effect because these doses of raclopride were all effective in suppressing the emission of ultrasonic vocalization during treatment, but after treatment discontinuation, it seems that this effect is lost because animals that were treated with raclopride emit a high number of calls after challenge. So it seems that this effect is observed when the drug is present, so when raclopride is present, so during repeated treatment, but is lost when raclopride is no longer present in the animal. Remember that these evaluations were performed 7 days after the discontinuation of repeated treatment. Whereas with the combination, so animals that received both drugs, you will see that the effect is retained because there is a complete suppression of ultrasonic vocalization stimulated by an amphetamine challenge administered 7 days after discontinuation. Another, okay, we also evaluate, this is the total number of calls. We also evaluated the emission of categorized calls. We focus on flat, trill, and the frequency modulated calls defined as the criteria proposed by Paul and this group. And we didn't see very much effects for the emission of Q-stimulated ultrasonic vocalization. We observed some effects for challenge, especially for frequency modulated ultrasonic vocalization. However, you see that the effect is in line with what was observed for the total number of calls. So SCH has some effects, whereas raclopride is basically behaves basically like a vehicle. So there is no persistent effect of raclopride on the emission of calls that are stimulated by a challenge. So another interesting point that was a point that raised some concern when these experiments were performed, and is a reason why I dismissed these experiments at the very beginning, is that about the 30% of the rats that received the combination of amphetamine plus either one or both dopaminergic antagonists emitted 22 kilohertz ultrasonic vocalization during treatment. And this, I mean, this is a strange finding because it led us to hypothesize that the animals had a dysphoric state during treatment. However, here I have to say that this emission of ultrasonic vocalization lasted for no more than 20-25 minutes, and that usually in our protocols we evaluate animals for 30 minutes to facilitate conditioning, because if we leave the animals for longer in the cages, conditioning may be somehow blunted. And it is possible that the animals had a transient and negative emotional state that was later overtaken by a positive emotional state, which may explain the presence of a high rate of callings in response to challenge. In any case, there was no correlation between the emission of ultrasonic vocalization during treatment and the emission of calls that were observed on re-exposure to the test cage or in response to challenge. So these correlations are not significant. And then, as expected, locomotor activity was suppressed by the administration of dopaminergic antagonists. We did this evaluation to clarify if there were no specific effects of antagonists on vocal behavior. However, the fact that animals emitted ultrasonic 22 kilohertz ultrasonic vocalization during treatment suggests that the effects that we observed were were not non-specific effects on calling, but they were effects that related to some neurochemical mechanism that regulates the emission of ultrasonic vocalization. So basically, the animals did not vocalize, not because their behavior was suppressed, but because dopamine is important for vocalization. So to summarize our findings, we found that the enduring increases in the emission of drug and cue-stimulated vocalizations that occur in rats repeatedly treated with amphetamine are attenuated by the individual antagonists of the D1 and D2 receptors and are suppressed by the combined antagonism of these receptors. These findings extend the previous results by demonstrating, as I was saying, that the simultaneous activation of D1 and D2 receptors is a requisite for the emission of 50 kilohertz vocalizations that are stimulated by amphetamine, even when the drug is repeatedly administered. So this dependence on dopamine activation can't be surmounted by the repeated administration of amphetamine, even if there are other mechanisms that regulate the emission of amphetamine-stimulated calls. To explain our data, we proposed a new hypothesis so that the concomitant administration of dopaminergic antagonists may or may not modify the affective state of rats that are repeatedly treated with amphetamine, depending on the dose of either antagonist that is administered and on the presence of an individual or combined antagonism of dopamine receptors. So we think that the individual antagonism of dopamine receptors may or may not suppress the positive effect that is elicited by amphetamine, whereas the combined antagonism of receptor does suppress the positive effect that is elicited by amphetamine. So this is what I was saying. And we also propose that dopamine D1 and D2 receptors synergistically interact to enable the full manifestation of amphetamine's persistent effects on calling behavior. We also propose that dopamine D1 receptors could predominate over dopamine D2 receptors in enabling the manifestation of the enduring changes in calling behavior that occur in rats repeatedly treated with amphetamine, because our results show that raclopride actually is not effective when an amphetamine challenge is administered. However, we should also consider that raclopride has also some activity on the D3 receptors, so we can't exclude that some different effects could appear if we use a selective D2 antagonist. And our findings could further elucidate the molecular mechanism and the neurotransmitter systems that regulate the emission of 50 kilohertz ultrasonic vocalization, as well as the significance of calling behavior in rats treated with psychoactive drugs that at least in part may be different from the meaning of ultrasonic vocalization in rats that are evaluated, for example, in a social context or in response to a non-pharmacological stimulation. So here I wish to thank people who participated in the study. These are people from my lab. This is a colleague, I don't know if somebody of you knows him, Emanuele Onaivi. He's from William Paterson, University of New Jersey. I mean, he was very important in this study because I decided to reevaluate the data after a discussion I had with him when I was visiting his lab in 2019 as a visiting scientist. And I mean, he stimulated some curiosity in me, and he prompted me to reevaluate the data. And so a take-home message for the young colleagues is that when you get some data that I mean, I'm not going to win any Nobel Prize, I think, but I can say that I really agree with Rita Levi-Montalcini that when you get some data that you don't understand, there is no useless data. So when you get some data that you don't understand, just put the data aside. And as you are seeing from my talk, you can reevaluate your data even a long time after the data was collected. At least I personally found this data interesting, so I hope it was also for you. And I thank you very much for your attention.
Thank you very much. So first question from the John, do you see any advantages of IP versus SC detection of amphetamine?
Oh, well, I see the advantage that the onset of the effect with the IP injection is more rapid. And I have to say that we inject amphetamine IP for a very practical reason, because in the first studies we did, we injected the drug IP, so we are keeping the same protocol. And as we say in football, a team that is winning should not be changed, so we don't change the protocol. But I don't know. I mean, we never compare the two routes of administration, so I mean, I can't really provide a convincing answer to this.
Thank you. So I'm not sure I got everything, but I was interested in the data about the V1 receptor antagonists, because we know that it also is involved in attention on processes. So I was wondering whether there could be a relationship between the data you showed and the modulation of attention in these animals.
Yeah, I think it's possible. Also, with regard to the Q-stimulated calls, maybe these animals were less attentive to the context compared to animals that received only raclopride. So yeah, I think that there may be definitely an involvement of the attentional process. Yeah.
So I'm a little hesitant to attribute your D1 drug effects to D1 receptors, because as you probably know, SCH222 although it is the most widely used D1 receptor antagonist, also has significant affinity for 5-HT2 and 5-HT1C receptors. And your doses were actually quite high compared to the rest of the
Yeah. I mean, the doses were high because especially the higher dose is very high. 1 milligram per kilogram is very high. But when the data were revisited, I decided to increase a little bit the dose, because we didn't see anything with lower doses. So yes, about the 5-HT receptors, yes, there may be a possibility that maybe. No, it had effects on when the drug was administered in combination with amphetamine, it blocked vocalization, but it had no effects on the long term. So, I mean, on the, let's say, acute, even though it's not an acute effect, I mean, when administered in combination with amphetamine, it blocked even at very low doses. I mean, like you just showed. But we did not observe any effects on the long-term modification, so we decided to increase the dose because of that.